Vanakkam. Welcome to this course on classics in neuroscience. In this video, we will continue our discussion of synaptic potentials and receptor potentials. In this video, we will look at other studies of sensory responses, because in the previous set of videos we saw receptor potentials. Before that we saw the work on aplasia and crayfish. Other studies of sensory responses started primarily with extracellular recordings. Remember, in the previous cases and in the previous discussion, we were looking at intracellular recordings. Here we look at extracellular single unit recordings. That is, electrode is not placed inside the cell, but rather in an extracellular matrix, and then the recording is done. This method was used by modern scientists and to analyze the sensory receptors performance. The early investigators was Edgar Adrian and Yngwie, I suppose that is how it is pronounced, Yngwie Zoetemelk, who in 1926 characterized the behavior of the sensory receptor using extracellular recordings and established a relationship between strength of the stimulus and strength of the response. One of the early studies. Later, this line of work was expanded for the visual system by Ragnar Granit, of course the famous Nobel laureate Ragnar Granit, and the basic response properties of sensory systems were recorded and documented by 1950s. For example, the Pacinian corpuscle and how it responds to stimulation were documented by this time. What are the limitations of this approach? One major limitation is the small amplitude of recorded potentials, obviously because these are extracellular potentials, and also you are not recording from a single cell. That means that you are recording whatever is the combined activity of multiple cells, or rather a population of cells, a summed electrical activity. So, it does not give insights into membrane properties and mechanisms like we have previously seen. The previous set of videos were all on how membrane properties are altered, or when you stimulate what happens to membranes and synaptic potentials and what happens. In this case, because you are measuring from a pool of neurons, from a group of neurons, you are not going to get insights about what happens in individual neuronal membranes. So, that is going to be missing. That is obvious, but it still needs to be stated. This kind of method was developed to study activity in the retina, a method called as electroretinogram, and activity from the olfactory epithelium. For example, the first development was on frogs in response to smell stimulus. This was called as electro-olfactogram, and this is used as a monitor of how effective a given stimulus is to given odor stimulus is by measuring this electro-olfactogram. Again, these are similar to modern methods used on humans, electroencephalogram, electrocardiogram. These methods do not measure activities of individual excitable cells, that may be brain cells or heart cells. What it is measuring is the summed field potential that is happening. Of course, the advantage is the ease of recording with large extracellular electrodes, so that is an advantage, but the limitation is the small amplitude. So, you have an advantage that you do not have to worry about intracellular recordings, you do not have to worry about micro electrodes and those. So, you can easily record with extracellular electrodes, but the disadvantage is also because of the advantage that because the electrodes are extracellular, there is the response is only small in amplitude. So, you really do not get any insight into the properties of the membrane or the synaptic mechanisms, the detailed mechanisms is something that you will not be getting. Similar studies were carried out to see how sensitive the visual system was, the hearing or the auditory system was, and the olfactory system was, and they found that these were indeed quite sensitive. They were almost at the theoretical limits. George von Bexey performed a fundamental analysis of how to mechanical stimulation cochlea responded. It was the first record of this kind of data. Of course, this was performed on cadavers, but despite that limitation, this continues to be a seminal documentation of the work on cochlea, for example. These are all the reference materials that we have used in the past 5-6 videos. For all these videos, these are the references. Please do check this. Some of these are Nobel laureates, for example, Torsten Wiesel here, Katz here, quite a few Nobel laureates here. Do check out these original papers. With this, we come to the end of this video. In this video, we looked at other studies of sensory responses, especially extracellular recordings of recordings, ERG, electro-olfactogram and other such methods that measured summed activity or field potentials over a relatively larger area, measuring the information from multiple neurons and not just single excitable cell. Okay, so thank you very much for your attention.